Good morning, guys, and welcome to our first recorded lecture. I hope everyone had a chance to look at the worksheet. If you haven't submitted that in the Dropbox, email it to me today, please. I hope it was helpful. You're going to need to know that process quite well. So know each step, know what happens at each step, and so on. For the beginning part of this unit, of course, you'll also need to be able to review the virus, the different shapes of the virus, and the structure of the virus. So don't forget to go back and review that stuff. Okay, so let's keep going with the unit. So pretty much for the rest of this unit, we're going to be talking about the influenza virus, which of course causes the flu. We'll talk about the structure, we'll talk about how it replicates, and so on and so forth. So we're going to have a look at the influenza virus, and you can see the picture right here. It's an envelope virus. It's also an RNA virus. When we're talking about RNA viruses, when RNA replicates inside of a cell, this way, it tends to make a few mistakes here and there. When those mistakes happen, we end up with the virus changing a little tiny bit each time we see a mistake. These viruses don't have a lot of structure, so they don't have a lot of complexity, so they don't have what they need, the machinery it needs to correct those mistakes. So as a result, we end up seeing these type of RNA viruses have a lot of mutation rates, high mutation rates. Now, this will change the virus quite a bit, and one of the advantages from the virus's point of view is if you have a bunch of different forms, you can respond quite quickly to environmental changes. So because of this, the influenza virus is quite robust, and it tends to last quite a long time. It's also an envelope virus. You can see it's got the lipid envelope that surrounds the capsid. On the surface of those envelope, we have two proteins. Now these proteins are antigenic, or we can call them antigens. And what an antigen does is it will elicit an immune response, or it will cause your body to have an immune response. So the two proteins we're talking about are hemagglutinin, which is what we often call the H spike, and another protein called neuraminidase, which is what we call the N spike. Now, hemagglutinin and neuraminidase are two structures on the outside of the viral envelope that help the virus do what it needs to do in life. One, it needs to infect a cell to get inside a cell so it can replicate, and two, it needs to get out so that it can go on and find another cell. So hemagglutinin and neuraminidase, they do just that. Okay. Hemagglutinin is a glycoprotein. It's on the surface of the influenza virus, and what it does is it serves as a receptor, or it's the place where the virus is going to join to the cell. Now, on the surface of most vertebrate cells, especially in our respiratory tract and our erythrocytes, the white blood cells, our cells will have a compound called sialic acid. Now, hemagglutinin will bind to sialic acid, and that is what's going to allow the virus to enter the cell. So the hemagglutinin allows the cell to be tricked, and then the virus is brought into our cells by receptor-mediated endocytosis. Once the virus is in there and it starts to replicate, it goes through the viral life cycle, it needs to get out. The neuraminidase is an enzyme, and what the neuraminidase does is it helps the virus get outside of the cell through budding, and it allows the virus to release itself from the cell, and then it can go on, and hemagglutinin can help it infect other cells. Now, what is one of the things we look for when we're talking about influenza is the fact that these two genes, the genes for hemagglutinin and neuraminidase, can mutate. And if they mutate and we see changes in these in hemagglutinin and neuraminidase, that's where we can see different flus season to season. So both of these H and N spikes, the hemagglutinin and neuraminidase, we can see variations in their shapes. So they'll have tiny changes in their shapes, but those tiny changes can represent a big difference to the virus. Those little tiny changes that we see that go from 1 year to the next year is what allows you to get a flu 1 year and then get a different flu, a slightly different flu, the very next year. So if you look down at the picture here, hemagglutinin spikes, the H, you can have 16 separate H types when we're talking about influenza A. The neuraminidase, the enzyme involved in the cell release. So these two things can change, and that's why when you see H1N1, we're talking about hemagglutinin 1, neuraminidase 1, H2N2, so on and so forth. Okay, guys, here's another look at the influenza virus. Again, you can see the envelope, you can see the neuraminidase. So we've got an envelope here. We got the neuraminidase, which is the blue arrow, and then the hemagglutinin, which is the green. We've got our sialic acid down here, which is going to be the receptor. So the hemagglutinin will bind to the sialic acid, and that will allow the cell, the virus, to enter the cell. Now, guys, I'm not going to show the videos that are embedded in the PowerPoints on these tape lectures in order to save some time, but this particular one here is a great video. You should definitely watch it. It will help you understand how a virus works, how it replicates, what's its life cycle. So I would definitely recommend you guys watch this one. Alright, so this is going to be a slide version or a cartoon version of the video that you just watched. This is going to show you the life cycle of a influenza A virus. We've got our eukaryotic cell, epithelial cell, which you'd find in your upper respiratory system, and we get our influenza virus. Our receptor on the outside is going to be the sialic acid, and the enzyme, or sorry, the glycoprotein on the outside of the virus is going to be the hemagglutinin. Will bind to the sialic acid, the receptor, and that will allow the virus to enter the cell by receptor-mediated endocytosis. So once the virus gets inside the cell, it's going to go through the same steps that we talked about when we were talking about viral replication earlier. So it'll decapsulate, or sorry, the capsule will come off. The genome will enter into the nucleus. We'll have transcription and translation. We'll get formation of new viral particles. Those virons will come together, and then finally the virus will be assembled. They'll be ready to leave the cell, and in this case for influenza, they leave by budding. So as they leave, they coat themselves in the plasma membrane of the existing cell, of the infected cell. Their membrane gives them the envelope as they leave, and the cell isn't killed when the virus exits the cell. So this is another slide, again another video. I'd have a quick watch if you wanted to have a look at it. It's kind of what we just talked about in the previous slide, except it's got some comments on it. Okay, so moving right along now, we're going to move into a topic in viruses which is very, very super important, and this is antigenic variation. And antigenic variation means how a virus changes so that it can cause illness in a person. So the antigen, which is the surface proteins that we talked about earlier, hemagglutinin and neuraminidase, these can change. And as they change, we can see tiny changes in the virus, which is something that helps trick our immune system so that we get sick every year. Or they can change a lot. And if they change a lot, that's the type of thing that can happen that can get us as a species or a local population into a lot of trouble with an illness. Two versions of antigenic variations are antigenic drift and antigenic shift. Now, antigenic drift results from point mutations of genes for the coding of hemagglutinin and neuraminidase. Now, that sounds like a mouthful, but I'll explain it on the next slide. Antigenic shift is when we get a reassortment of viral genes. So this is a much bigger change in the virus, and this one, antigenic shift, can cause us a lot of different problems. Again, another video explaining antigenic drift, so watch this video, please. Okay, antigenic drift. When we're talking about antigenic drift, what we talk about are tiny mutations in the genes that code for the hemagglutinin and the neuraminidase, so the receptors that we see on the outside of the virus. Now, if you look at the picture here, we start off with a flu virus, and it goes through an antigenic shift. And if we look at the difference between the spikes on the outside of the new virus and the old one, we see that they're slightly different. Okay, they're not pink like we started off. Now we've got yellow and pink ones. We've got a bunch of different colored ones, green and pink, slightly different shade of green and pink. But those little changes represent tiny, tiny changes on the hemagglutinin and neuraminidase on the outside of the virus. Now, those little tiny changes, those are enough to trick our immune system. So our immune system doesn't recognize the virus that we had last year because it doesn't look exactly the same. It's not the same one with the pink spikes. Now we've got a similar virus, but it's got yellow spikes. So our immune system doesn't recognize it. It doesn't protect us from it. So antigenic drift is what causes people to get the flu each year, or even twice in the same year. I mean, that would be a bad year, but it could happen. Those little changes are just enough so that the virus can fool our host antibodies. The antibodies won't recognize the viral cells, it won't attack them, and that allows us to get sick again. Now, antigenic drift is what can cause flu epidemics, and that means a flu that's really bad in a certain area. If you visited your cousins, for example, in Kuwait or Saudi Arabia or somewhere, they could have a very bad flu that's there, but it doesn't travel as far as Qatar. So you get changes in flu. It's not a massive change, but it is enough to make you sick year in and year out. Antigenic drift is the reason why we have different flu vaccines each year. We don't know what the flu is going to be in the next year, so we give you our best guess from what we had the previous year. And that's how we formulate our flu vaccines. Now, this figure here is important because it highlights what a point mutation is, why we get changes in our the H and N antigens, but it also explains to you how the flu shot works. So if you read down through these steps 1, 2, 3, 4, 5, 6, it'll explain to you how we put together our flu shot, and how the flu shot will help your immune system become prepared for the viruses that we've seen last year. Now, what we notice is interesting is if we look at the picture between 4 and 5, if we look at the, one second, sorry. If we look at the different spikes that are on the outside of these two viruses, we notice that they're slightly different. In this one up here, we've got the blue club-like shape, and on this one here, we've got kind of a purple heart on the end of it. So that little change right there represents a point mutation. So somewhere along the genes for